What do you notice about them? They start with? Starts to slant, so it doesn't make a particularly pretty straight line, does it? In fact, having that molecule in front of you might indicate why in 3 dimensions it's not a ladder. Remember, we said a ladder isn't accurate. What is? Staircase. The molecule itself, because of its shape, does not make a pretty straight line. It starts to bend by itself. So it's not straight. What's on the edges? So there's a sugar and a phosphate on both sides, right? So both sides have a sugar phosphate backbone. That's what we call that. What else do we have? Not only are A and T complementary and G and C complementary, a scientist whose I think his first name was Edwin, but his last name is Chargaff. Edwin Chargaff told us not only is it that they are complementary, that the amount of adenine equals the amount of thymine, and the amount of guanine equals the amount of cytosine. And we call that, because of his contribution, I have no idea why I dotted my R, Chargaff's rule. So that's his rule. A equals T, C equals G. The amounts, percentages specifically, are what he used. Looking at it, what's in the middle? So if the edges are sugar and phosphate, what's in the middle? The bases, nitrogen bases. So the middle is made of rings of nitrogen. How many rings across is each step? So as I go across, if I add up the number of rings in A and T, how many rings is that? How many? 3. And if I add up the rings in G and C, how many is that? 3. So the complementary base pairs mean that it's always 3 rings wide. The spacing is always the same. 3 rings wide at any given point. Not anything that's bothering you about what's on your paper? Mm-hmm.
Well, on our paper, it would only appear that there's 2.
Depending how you lined it up, absolutely. Do that again. Compare what I have here in my picture to what you have in yours. This is organized in your picture. Not only is there something not organized about your picture, but there's something visually upsetting.
Is it that they all don't fit together perfectly?
In reality, they do fit together perfectly. They will.
On this paper.
And in your paper, if you didn't do like a very precise trimming job, they won't fit together perfectly. Catherine? Oh, sorry. Yeah, half your paper is upside down, right? Like if I handed you a photocopy where half of the page was upside down, you would look at me like I was an idiot who didn't know how to use the photocopier, right? Right? You'd be like, yeah, clearly she failed photocopying class in the School of Education. Listen, they should teach it, but they don't. Half of it's upside down. When we talked about the structure of DNA being a double helix with 2 strands, we said the strands ran anti parallel. And we really didn't like the idea of anti-parallel when we first learned it. We're like, I don't understand what you're talking about. But as soon as I gave you pieces and told you you could do whatever you needed to do to orient them in space so that the nitrogen bases were touching, you simply picked up one piece and turned it upside down, right? And some of you called me over and you're like, no, if I have to do this, then it has to be upside down. Like that violated your sense of being because the letters were upside down, weren't they? Don't worry about that. That's normal. It should happen. That's what we mean by anti-parallel. One side of the strand is not the mirror image, but rather the inverse of. It's upside down. That's anti-parallel. On my picture here, you can see it not so obviously because these fool textbook writers, they made the letters all face the same way, right? Apparently they didn't want to get a bunch of phone calls about bad copies in their bad pictures in their books. But you can tell because oxygen on this side is pointing up and oxygen on this side is pointing down. That's anti-parallel. It matters because if they weren't anti-parallel, if I took this and flipped it over, that's not what the molecule's shaped like in 3 dimensions. That's not what it looks like. The molecule doesn't exist. And if I did do that and I took that and I flipped it over, it wouldn't line up to make those pretty red lines that you so kindly told us about being in the middle, right? You said there's bonds in the middle. If you change how they're oriented in space, they can't bond. So being anti-parallel is absolutely essential to the shape of this molecule. Has to be. Now I'm going to blow this one up so we can take a look at it so you can see the bonds a little bit better. Something tells me that maybe on paper, as AJ so kindly pointed out, they don't fit together so perfectly. Is this accurate? Yeah. The day I learned that I could like shrink stuff that I wrote was like the best day ever. It was last week. The best day of my life was last week. All right. Is that big enough for you to see it? I'm going to change to a happier color than these. Green is happy. And that red and pink is happy. So because of the anti-parallel nature and the way that these fit together like puzzle pieces, it lines up certain atoms with other atoms. You'll notice that I have a red dashed line here and a red dashed line here. Those represent a special kind of bond you heard about in Chapter 2 called a hydrogen bond. And we always indicate hydrogen bonds with lines that go like that rather than a straight line. Because they're more like two magnets when they stick together. You can separate two magnets, right? You just pull them apart. These, same thing. I can take them, I can just pull them apart. It's not really that difficult. But when you get the magnets close enough to each other, even after you've pulled them apart, what do? Snap back together. Same thing will happen here. If these are like remotely kind of close to each other, they snap back together and those bonds form again. It's because oxygen and nitrogen love electrons. They are electron hogs. Hydrogen, which makes them slightly negative. Hydrogen is slightly positive because hydrogen's not really that concerned about electrons. It's like, whatever, you take that one, doesn't bother me any. So it's slightly positive. That attraction makes a bond here. So hydrogen bonds occur between somebody who really loves electrons. Nitrogen, oxygen, and fluorine are the usual suspects. And hydrogen on a molecule next to it. So that's not the same molecule, it's a different molecule. That's a hydrogen bond. Between guanine and cytosine, we have 3 because I've got my big old hog oxygen right there. It's being an electron hog. And it's right next to hydrogen who's like, It's okay. I don't mind. So they're attracted to each other. Same with that nitrogen and hydrogen, and that hydrogen and that oxygen. If you swapped, say put cytosine next to thymine, you would have this oxygen next to another oxygen. You ever had two exceptionally selfish people in one spot? What's it like?
Not good.
Not a picture of harmony?
No.
What tends to happen?
There's fight, they fight.
There's tension. It's awkward. You have two people who are demanding attention or demanding objects. It doesn't work. They don't complement each other very well as far as personalities go. So that's what happens here. So thymine and cytosine will never pair together. Same problem with guanine and adenine, except instead of having two incredibly selfish things next to each other, you'd have two fairly laissez-faire things next to each other. They're like, no, you go ahead and take it. Oh, no, you do it. No, it's yours, really. Oh, no, you decide. You ever been in a room with two people who can't make a decision? How much fun is that? How many of you are like, finally, you just make a decision so you don't have to deal with them? Right? So we can't ever have that happen. It doesn't work. They don't complement each other well. That's part of the reason this anti-parallel thing is such a big deal. If we label it, maybe on your diagram you noticed, your original diagram started with the 5 at the top, right? You had a 5' on one side, and it ended with 3'. Directly across from 5' should be a 3'. And across from it is a 5'. Why? Because this is upside down. That's all. It's not as bad as the first day we looked at anti-parallel, right? You're like, oh, okay, with like things in 3 dimensions. All right, fine. You, my friends, have 6 billion of those things that you made 8 of in every cell. Not in your whole body. I mean, every cell in your whole body has 6 billion. So remember, like, way back when in the cell cycle, also known as, like, last week, we said that DNA had to make a copy of itself during synthesis. Every time any cell divides, it needs to copy all 6 billion. How many of you are like, yes, I could copy a 6 billion page book with complete 100% accuracy? No one signing up for that? And I don't mean like take it to the photocopier. Oh no. I mean you sit down and write letter for letter all 6 billion. No? How many of you are like, I would make a mistake? No. Unless you have the hubris of our friends Daniel and Robert.
I am their Hebrews.
Well, that's good. Do you know what the downfall of Macbeth was?
Did he die?
Yeah, you know why he died?
Because he was perfect.
No,
He was unperfect.
because he was proud.
Proud of being perfect?
No, his hubris was his tragic flaw. For those of you, when you get to tragic flaws and dramas, remember, you heard it here first. What was his tragic flaw? His hubris.
What is a Hebrews?
I just don't make mistakes.
I just wanted to be part of their Hebrews.
Hubris is pride or arrogance.
Oh, I'm not really like arrogant. I'm just trying to say that I don't make mistakes.
I'm not arrogant. I'm just saying I'm better than you.
No, I'm just confident in my perfection.
Which is precisely what Macbeth would have said before he died. Yes. Let that be a life lesson.
I'll see.
When do you guys read that?
Next year. I saw that in Jimmy Neutron.
What?
Oh, yeah.
It was,
I don't
it was
remember
they had
that.
like a play of it.
Dude, Jimmy was my boy.
Yeah, I know. No, it was Carl.
Green
Oh,
blood.
Carl? No, wait, Shane.
Shane.
All 6 billion of your nucleotides
Yeah,
is called your genome. So every single organism has a genome. It's every single nucleotide that it has. So a genome is every. And we're going to underline it in green. Every nucleotide in the cell of an organism in order. That's your genome. So your 3 billion base pairs, because remember we said it's 6 billion total, so since they're paired, it means a strand of 3 billion in order. That's your genome. Now order here matters. Here's why. That's what makes us unique. So human beings, we've got 3 billion base pairs, right? We all have 3 billion. Every single one of us. It's the order the A, T, C's, and G's occur in that make us different. So I'll give you some examples of things controlled by the order of A, T, C's, and G's. Your hair color, your eye color. We've already discussed whether or not you pee pink when you eat beets, right? That's controlled by the order. Whether or not you
What?
can... Hey, you can do it too. That means ours go in the same order. Any of you who can't do that, don't worry about it. It's not a flaw. You are not broken. It just means your nucleotides go in an order that prevent you from doing that. In which case, Robert will laugh at you in a way that says, ha ha. Like Nelson from The Simpsons.
Wait, how can you not do it?
Yeah, honestly, how do you not do
Hers
that?
is in half.
My mother cannot do it.
Really?
Yes.
Okay, how does this do? Some people can't do this.
So the order that you go in affects the proteins that you make. Some people, their proteins prevent them. Just like whether or not you have that dent in your chin called a cleft, just like whether or not you have a widow's peak, all that is controlled by the A, T, C's, and G's in the order that they go in. That's your genome. Every single one of those.
Wait,
In order.
like,
Don't scramble them. Don't like drop them and have them like spill all over and then pick them back up.
So if you have like a dent in your like in your chest or something, then like your chromosomes go in order.
what do you have in your chest?
Usually,
Yeah, I know someone.
usually that's caused in gestation. So when the bones are forming is typically when that happens, which is not controlled by genes, but rather caused by something that happens in gestation. Somebody last hour was like, well, can you ever change the order that yours came in? Yes, but you don't want to. You don't want to change the order because changing the order usually ends up causing this disease called cancer. I don't know. I think that's bad. What kinds of things cause changes in the order? Anything that we call a mutagen. So, you go to the dentist, they lay that big lead vest across your body when they expose your face to x-ray. They're trying to protect the rest of your organs from x-rays because it can cause mutations in the DNA of the rest of your organs. So they're just looking out for your liver and your kidneys and all the other stuff, I heard somewhere they do jobs or something. So they're just trying to save you. That's why. You also ever wondered? They put that thing up to your face and then they leave the room. It's okay to do it to you, but they're getting out of Dodge. Why? Because they do that probably like 30 times a day. You get one teeny tiny dose every year. You only get one set of x-rays when you go to the dentist, right? Once a year. You go for recare 6 months, but once a year you get those x-rays. One little dose. They do that every day, 30 times a day, for their career, so 5 days a week. Yeah, they leave the room.
Wait, so it's like radiation, right? What if you just left it on? Are you done?
Yeah, I'm pretty sure you're done.
So, yeah, you
Guns
probably
up,
done.
guns up.
Wouldn't it not matter if you had it over the rest of your body because it affects like a certain cell it would just strength?
Yes, but the dose that they give you, where they give it to you, is in such a small amount that it's not likely to metastasize. They would be more worried about it if it hits lymph nodes. So you know where your lymph nodes are? You've got like all kinds of crazy sets of them, right? Like here and here and here. They're all throughout your whole body. If they were aiming at your lymph nodes, they'd be really worried, but they don't hit those. If they do hit those, you have a particularly bad hygienist or doctor. They don't pass dental school. You don't have to worry about those.
My bad, my bad.
You're not perfect.
I'm not, actually, yeah. I'm sorry. That's pretty dark.
But sometimes when I think that the only things you've learned from Jimmy Neutron, I'm like, I'm just gonna go home. Like I'm just gonna sit on my couch and eat Cadbury eggs and just be.
What are cat? What are those?
You know, the ones in the purple bag, by the way, not like the gooey ones. Those are disgusting. The ones in the purple bag. You know the ones. They're like pastel in color.
Wait, they're
Everybody
not,
waits for
wait.
Easter because they're better than
Like
all the other
chocolate
candy.
eggs or regular? Wait, what?
How do you not know?
Alright, never mind, never mind. Chocolate eggs. I am.
Yeah! Yeah, what he said.
I am.
So, how do we make sure all 6 billion get copied? There's got to be a method. There is a method. It happens during the S phase of interphase. And then think about that circle again. I said you naturally interpret it as a pie chart because some teacher taught you so very well. So you've got this nice little pie chart. Was it like half of the pie chart that was synthesis? 75%? Was that S? How about 33%? A quarter of it was synthesis? It's probably somewhere around a quarter of that wedge was synthesis. It's not very much. So not only does your body have a way to do that, it does it quickly and error-free, mostly. Errors happen, but most of them get caught. Are you reassured? You're like, no. Well then, put on sunscreen, avoid high doses of X-ray, and avoid anything termed a carcinogen. You'll be okay. Oh, and eat well, exercise, hydrate, and sleep enough. This sounds like stuff you're supposed to do anyway, right? And don't eat like lead. Don't eat paint chips. I know, you're like,
That's
oh,
probably a good idea.
that was my snack I packed before track practice. I was gonna eat some paint chips.
Wait, what if you actually?
Last page of notes I'll have you copy down. DNA synthesis is also known as replication. The process is semi-conservative. Semi meaning half or part. Conservative meaning not like in disposition, but rather saving. When we conserve something, we save it. So it's half saved. We learned that from an experiment we call the Messelson-Stahl experiment. I'll begin tomorrow by showing you a video about the Messelson-Stahl experiment. But basically, it's STHL, by the way, STAHL. The Messelson-Stahl experiment basically changed the replication environment of bacteria so they could figure out the mass or the weight, the density of new strands of DNA.
Where should we?
For you is that it's different between prokaryotes and eukaryotes, right? And I said that's because prokaryotes were a single circular chromosome. And our eukaryotes were multiple linear chromosomes. Exactly 46 if we are discussing a human being. I've only drawn 2 for sake of expedience. One of the major differences is the number of origins of replication there are going to be. So 1 origin in Prokes, multiple in Eucs. Remembering that prokaryotes are, in fact, all of the bacterial kingdoms. Eukaryotes are the everybody else kingdoms, fungi and Animalia, Protista and Plantae. We had some confusion in the last test where some of us called Plantae prokaryotes, and I was a little bit worried about that. Prokes are only the bacterial kingdoms. Replication is going to start at the origin of replication. What a better place to start than, the beginning. And it's going to move in 2 directions on the open strand. So here we have our before picture showing the origin. And this is the during picture where the origin is going to open up and it's going to move both ways at the origin. So it goes both the left and it goes to the right as well. So it goes left and right. That creates what we call a replication bubble. It looks like a bubble, right? It's like a little blip. It's a tiny replication bubble. And every replication bubble is composed of 2 replication forks. Here's one fork, and here's the other fork. So every bubble has 2 forks. I think the next slide is all of the major players. Yep.
Where are the 2 forks?
So here's one fork, and here's the other fork. So one side is a fork, and then the other side is the second fork. These are all of our major players involved in DNA replication. I'm going to give you a moment to just write down their names. If you want to write down their name and write them in a list numbered 1, 2, 3, and 4, starting with topoisomerase, helicase, primase, then DNA polymerase. So give yourself some space to write their functions. As you wrote down the names of these things, there are some things you should have noticed. All of them end in ACE. In biology, that's true for all of our enzymes. Enzymes are just biological catalysts. If we remember, a catalyst is anything that speeds up a chemical reaction but doesn't become part of the product. So all of these things are simply designed to make DNA replication go faster, but they are not part of the DNA after it gets made. So they show up, they build it, and then they leave. Remember when you were a kid and you would sit on the swings at the park and you would twist the swing in a circle and it would make the chain of the swings curl up nice and tight just so you could swing and just spin in a circle? You put a certain amount of torsional stress on those chains. And that word torsional stress, you understand what it meant because the tighter you got the chain, the faster you spun when it unfurled, right? So that's because of all the energy that's bound up in that twisted physical structure. DNA is a twisted physical structure. And if you try to uncurl it like you would uncurl a slinky, if you tried to uncurl it like you uncurled a slinky, if it causes more stress farther down the chain is going to break. Now, if this were true, we could never uncurl DNA to copy it. So this first enzyme up here, topoisomerase, its job is to release the torsional stress, that twisting tension on the unwinding helix. Your textbook shows it as like this strange, I don't know, like, I think it's red thing that fits around the helix and moves in either direction. The next enzyme who's involved here is helicase. Helicase's job is to cut the hydrogen bonds in the double helix. So it cuts those hydrogen bonds in the helix. But the problem is, you remember I told you guys when we looked at that structure, I told you that those hydrogen bonds automatically form if you get nucleotides close enough to each other, right? You get them nice and close, and they automatically form like two magnets that get stuck together. If I separate two magnets, which is what helicase would be doing, separating two hydrogen bonds, but I leave the magnets close enough to each other, what will they do? They re-stick, right? They automatically go back together. So if I don't attach these single-stranded binding proteins to the unwound and opened helix, it just sticks right back together. That's not helpful. So those single-stranded binding proteins attach and keep the hydrogen bonds from rejoining or reforming. Did you get the name of those things written down? There were some people whose pens didn't move, and that concerned me slightly. What was the name of my SSBP?
Single-stranded binding proteins.
Excellent. They prevent hydrogen bonds from reforming in the open helix.
So after the helix is separated and it's like forming another fiber, do they just leave?
Mm-hmm.
What is the BP stand for?
Binding protein. All of these ACEs, by the way, they're all proteins. Anything that ends in ACE, you should anticipate being a protein. Primase. Do you guys ever painted a wall before?
Yes.
You have to put primer on the wall before you paint, right? What's the function of primer? Yeah, it gives the paint something to stick to. If you don't give the paint someplace to stick to, it actually absorbs into the drywall, and then you have to put up a whole bunch of coats of paint because drywall acts like a sponge and just sucks up the paint. Well, then you have a really, really bad paint job. You've wasted tons of money because paint is not cheap. Seems silly. So we attach a primer so paint has something to stick to. Just like primer, in that case, nucleotides can't stick to nothing. So on the new strand, there has to be a small, short primer that gets attached. And then new nucleotides can be added. That primer is attached by prime ACE. You'll notice that obvious. Primer is attached by prime ACE. The helix is unwound by helicase. So that's generally how we name things. It tells us what our next enzyme does. A polymer is made by DNA polymerase. It attaches complementary nucleotides to the primer. I should tell you the primer, by the way, is made of RNA. It's not made of DNA. It is an RNA primer. DNA polymerase has another tiny quirk, other than not being able to attach to nothing. When you read a sentence in English, what direction do you read? Left to right. If you attempted to read a sentence from your textbook from right to left, will it mean anything? No. You're not going to get anything out of it unless it's a palindrome. But how many sentences are palindromes besides, like, all I can think of are words like race car and auto, Hannah. Is it even a thing?
Yeah.
Like race car is at least like, okay. doesn't love a palindrome? We don't have all that many palindromes, and much like that, DNA polymerase is very limited. It's a little quirk. It can only read the template strand in one direction. Now, your inability to read a sentence in English from right to left, how many of you feel as though that's a flaw? You're like, clearly I'm a failure. I can only read in one direction. None of you? You don't feel like you've somehow like failed at life?
I can read it too.
You can read in both directions?
Yeah, I guess.
Didn't we discuss hubris yesterday?
Well, I'm doing it right now.
Japanese books are in the other direction.
Yes, there are other languages that do go in the other direction, but that's why I specifically specified English, right?
Yes.
So none of you feel like a failure because you can't read in the other direction? So we don't refer to this as a failing on DNA polymerase's part. It's more of a quirk. It can only read the template 3' to 5'. It's not its fault. It's just the way the language was written. It can only read the template from 3' to 5'. Now we know who all the players are. We know what everybody has a job or function here does. We have to see how they do their jobs. We actually learned them in order. Topoisomerase has the first job, then helicase and single-stranded binding proteins have the next job, then primase has the next job, then DNA polymerase has the last job. So that's the exact order that they're going to be introduced in. And we're not going to look at a whole replication bubble. We're going to look at a single replication fork. And then we're going to see the whole process in motion. Now, there will be tons of questions you have as we learn the process not in motion. Feel free to ask those. That's totally fine. I'm happy to answer them. Some of them will become a little bit more obvious once we see the whole process moving. So we'll do this, then we'll video it so we can see it moving, and then we'll see what it actually looks like in real life, like for real, with a marvelous computer generated. Original strand, shall we make it red? Or like magenta reddish?
I'm feeling red.
If the top strand goes from 3' to 5', what's the bottom strand? 5' to 3'. I probably should have written that 3, like, up there-ish.
So this is like the original DNA.
Mm-hmm. So here's our old one, the one that we need a copy of. And you'll notice that its origin of replication's already been opened up. So we have a replication fork here. So we're going to have our topoisomerase down here doing its thing, releasing torsional stress so the whole thing doesn't like turn into a giant tangly knot. And we'll label that with a nice T for topoisomerase, so we know exactly who that is. And then here we have our friend helicase. Helicase is attached and is doing its job. Helicase is there. I'll use that one. Cutting all those hydrogen bonds. So snip, 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 and all the hydrogen bonds in between those bases. Primase, well, single-stranded binding proteins. We'll make them this color. They just attach right out here, and they prevent the whole thing from coming back together. Who comes next? We're doing them in order. We did topoisomerase, helicase, single-strand binding proteins. Angela, who's next? Primase. Primase floats into the picture and is going to attach a nice RNA primer at the 3' end, right? So right here. Here's my RNA primer attached by primase. I'm going to label primase with a lovely P. primase attaches a primer. Who's the next enzyme? Angela?
The DNA polymers.
Mm-hmm. So here's our friend DNA polymerase. If DNA polymerase encounters a cytosine, what's it going to attach to complement it? It's going to attach guanine. And then if the next thing it encounters is an adenine, what will it attach? It's a thymine. So DNA polymerase, my enzyme, is just going to keep moving that way from 3' to 5', attaching complementary bases as it goes. Helicase is going to keep going this way. Just keep following topoisomerase. It's going to keep unzipping all the way down the strand. DNA polymerase is just going to keep on following, attaching complementary bases. That's not so bad, right? Like, yeah, okay, fine. We unzip it, and then we just attach complementary nucleotides.
Does the primer go before the DNA?
Mm-hmm. Yep, so primer would have attached here, and then DNA polymerase attaches, and then moves that way.
So is the DNA after the primer?
In sequence or time. Like, it comes before it in the strand, but it comes before it in time too. If this is the beginning, if this is the origin, you have primase, and then we move this way down the strand. And we're just going to keep going that way because we're reading from 3 to 5. That's our template from 3 to 5. DNA polymerase totally happy.
What about the other side of sugar?
We always like to know there's always fine, okay, I get it. What about the other half? This half is super fast. Leads the way, it's why we start with it. It leads the way, it blazes trails. We call it the leading strand. And on that leading strand, replication is continuous. It's just going to keep replicating. Just going to keep going. It's going to keep going. It's going to keep going until it encounters another replication bubble that was down the strand somewhere. It's also replicating. Once it encounters one, it's done. This bottom side is not quite so simple. DNA polymerase can't attach here and read that way, right? It's not allowed to read in that direction. So what primase is going to do, primase attaches here on the 3' side of this open strand. So here's our friend primase. It attaches our RNA primer. And then a new second additional DNA polymerase attaches. And it's going to continue reading this way, attaching complementary base pairs. As it moves down the strand. So it has to move in the opposite direction. Why? Because the strand's oriented in the opposite direction. How many of you are like, I thought you said it was going to be complicated? That's not that bad. What's helicase doing? It's moving this way. Our primer attached here. DNA polymerase is moving this way. So now there's a whole new open chunk of unreplicated DNA, right? You're like, what the heck? It's okay, no big deal. Another primer attaches as close to it as it can to helicase. Another DNA polymerase attaches, and then it works backwards until it encounters this primer. So it just has to do it in little individual pieces. The more that opens, then a new one attaches and works back. While it does that, more was opening, so another attaches and moves back. So it works in fragments, and those fragments have kind of a strange name. We call them Okazaki fragments. They're named for the scientist who discovered the fact that this is how it had to have happened for any of this to make sense. And this lags behind in speed because doing it little chunks at a time takes a lot longer. So we call this strand the lagging strand. So here's our lagging strand. And we can't call it continuous replication because, well, it doesn't continue in a straight line. So we call this discontinuous. So the lagging strand is discontinuous.